Dear colleagues, dear guests, we are sorry a bit for this delay, but I think we will skip these opening remarks by by myself. And we, in our agenda, we have four talks, four 15 minutes talks before coffee break. And I would like just give the floor to Professor Nicotera just for the first contribution research on neurodegenerative diseases in Germany.
Thank you very much, Professor Sergeyev, and thank you for the hospitality once again. I will introduce you to the German Center for Neurodegenerative Diseases. This has been, probably is the largest organization in Germany dealing with neurodegenerative diseases. It is trying to address a problem which is becoming a global threat to our society. So if you look at the data from the US in terms of causes of death between 2000 and 2015, you see here that whereas many diseases, including different types of cancers or HIV, are in decline, the deaths because of neurodegenerative disease are actually rising significantly. And of course, this is a problem linked to age or to the aging process. And of course, the older you are, the shorter is the lifespan you expect. In Germany, we expect now a doubling nearly of the disease in the next coming year up to 2040. And of course, because the demographics are changing, so in this panel here is basically the distribution of age in populations at different ages, 2000 and I can't even read here, I think 3 and 2013, sorry, it's a bit too small. And of course the risk for this increases, and significantly this will increase primarily in countries where the life expectancy is increasing much faster, and Russia is one of those. China is another one of those, and therefore the implications of dementia for society are actually even more severe in these countries. Which are the challenges for neurodegenerative diseases? Well, first of all, we don't know the causes and mechanism of diseases, and therefore we need to understand these causes of neurodegeneration, and in particular the role of the immune system, the immune response and inflammation are becoming more and more central to understand how the disease manifests. There are complex interactions between the genetic and environmental factors. In addition to the normal aging process, of course, the understanding of aging per se takes a very relevant role in this disease, which manifests at ages above 85, particularly for dementia. Parkinson's disease is a different distribution, but is increasing in parallel as well. And in fact, many cases of dementia are actually Parkinsonian syndromes, which then tend to evolve into dementia. And of course, we have a number of different diseases. So one of the problems that we have, it is that diseases are not well stratified. So we don't know exactly how to treat different diseases because we don't stratify them very well. We need to characterize diseases much better. There are not too many models that can realistically mimic human disease. And the reason is that the animal models that we're using often mimic only one particular feature of the disease, but not the entire pathological process. Also, neurodegenerative diseases tend to develop decades before the symptoms are manifested. They can have multiple entry points, mixed forms of dementia, overlaps of symptoms, which makes diagnostics, therapeutics, and in general, trying to be able to have also non-pharmacological interventions very difficult. And to date, we have no disease-modifying therapies. This is a major problem in neurodegenerative diseases, a difference with other conditions where we can at least have successful symptomatic therapies. Here we barely have nothing to be able to treat the symptoms except some form of psychosis or mild kind of improvement. So the German government in 2009 decided to create a national center to be able to address the problem of neurodegenerative diseases. And we were funded with a unique structure. So we are part of the Helmholtz-Gemeinschaft, so the Helmholtz Association of Research Centers, which is one of the 4 core-funded organizations by the federal government in Germany. But we're also cooperating very closely with several universities at 10 different sites. The main site is in Bonn, and in Bonn, of course, we cooperate with our neighbors in Cologne, in Jülich, but we also have a spreading of the dysidenia at different sites, and at each site we cooperate with one of the major medical faculties and universities. So this facilitates the possibility to create multi-center clinical studies. We share the infrastructure, and of course we join the policy of the universities in appointing top scientists all over the world. So in 2009, we were barely a few tens of people, and now we are over 1,300 employers in the whole country for dysidenia, and many of which share a joint appointment with local universities. So our mission is to understand the causes and find preventive and therapeutic strategies against neurodegenerative diseases. And the idea is just say this, to bring under a single organization the best of German research on neurodegenerative disease, from very fundamental research to human studies, including population studies, clinical and health care research. And by doing this, we try to break down barriers between the different research areas by bringing scientists together and try to be able to cover the whole spectrum of translational research. So if you really look at how we are organized, actually we don't have really barriers between the different topics that we address, but we have generally an emphasis on brain function, pure brain understanding of brain function, disease mechanism, clinical and healthcare research, and then disease prevention and healthy aging. And you can see here across the different areas, different sort of transversal topics that we can address, like genetics, epigenetics, neuroinflammation, or data science, for example. The target of our translational research, more oriented to human studies, is also to be able to enable clinical trials or clinical interventions. And because we try to stratify patients in a much more efficient way, that means that we have less patients at each site than you normally would expect. So we try to characterize them in depth. So to try to be able to address the possibility of studying individuals, but also doing clinical trials, what we are doing, we are trying to create cohorts. So our attempt has been to create different cohorts for different diseases where patients are highly characterized. We have about 10 cohorts now for different diseases, including rare disorders. And then we can do with these cohorts phase 1 and phase 2 clinical studies. We have already a number of interventional studies, but we also try to understand, for example, what is the transition between a subjective memory impairment and the real disease that it manifests. So this is a clinical research platform, which is formed by 12 clinical centers at 10 dysidenia sites, has common standard operating procedures. It enables national clinical trials. And of course, it also tries to innovate in terms of new outcome measures. This is another problem in neurodegenerative disease. How do you figure out if a patient responds to a treatment or not? Usually you run cognitive tests, which are okay, but they are not the very best way because the noise in these cognitive tests can be very high. So we need much more much stronger indicator of outcome. And one of these is, of course, imaging, and you will hear from Catherine a lot about this in her lecture, I suppose, but we have been trying to uniformly harmonize imaging throughout our sites. We have available in our centers 3 Tesla, 7 Tesla, MRI, MR PET, and of course we link this to cognition science, so we can try to be able to provide better outcome measures. At the same time, we also use molecular imaging to be able to understand the validity of new targets and the interaction of neuronal networks. Finally, we dedicate quite a lot of research into healthcare, but also in population science. In population science, we have created a cohort that aims to recruit about 30,000 people in the Rhineland study that is followed throughout their life course, meaning that these individuals are observed with a period of 3 years or 4 years. And instead, with this any healthcare research, we try to be able to improve the quality of life of people who are already affected by dementia. And in this direction, we have created a new figure in the German healthcare system, which is the dementia care manager. We have actually created also a curriculum for that, and qualified nurses, the dementia care managers, play a key role in the well-being of people with Alzheimer's disease. And the reason is that they can follow the patients at home, they can help them if they are hospitalized by helping the carers to be able to understand the conditions. Try to imagine an Alzheimer's patient with a broken hip going to the hospital, very difficult to deal with it. So the care managers are actually having a very important function in the care plans. And also we use, of course, IT-supported care plans for the patients. And we're trying to be able to extend this from the initial studies in the north of Germany to the whole country. The other thing we're doing is obviously to develop new drugs. And we felt that we had to use a slightly different way by doing this, by attacking this task. We're very good at discovery, so basically we can identify targets very easily. The problem is that when we cannot develop them without the contribution of the pharmaceutical industry, so either we go the traditional way, which means individual cooperations, or we decided to go in a different way to be able to make a strategic cooperation. So the first partner was Orion Pharmaceutical from Finland, and together with them we developed a new way by which we jointly collaborate by investing into the drug development, which means for them, they don't have to take care of the initial part of development, and we don't have to take care then of the proof of concept in the sense of the ADME toxicology and the identification of new lead compounds. So we screen compounds together with them. Eugenio Fava, who is here with us, is the leader of our core facility doing that. We try to be able to cooperate by interactions or iterative interactions in trying to understand which are the best hits to be able to develop the lead compounds, and then eventually industry will synthesize enough for us to be able to go back to our clinical infrastructure and do the phase 1 clinical studies. We share 50-50 of investment and ownership. And this actually was nominated for the Industry Scripps Prize in 2016. And since then, we have extended this model to ChemDiv, which is a company based in San Diego, but with a lot of Russian colleagues working in it. Many of you may be familiar with it. And with Eisai, which is one of the major Japanese companies developing drugs for neurodegenerative diseases. The other challenge is data science. We have touched upon these actually quite extensively in our discussion already this morning, and you all know that you have a tremendous input of data in clinical and fundamental biomedical research, from genome sequences to different type of data sources from clinical environments, from diagnostics and therapeutic, and from a model system and prognosis. So how do we cope with that? Well, one way of coping with this is, of course, to be able to store them in servers and servers and servers, but the problem is that we have to exchange data. So we don't want to keep them there. And to exchange data, then we, of course, need new ways by which we can attach the problem of computing. And so we partnered with Hewlett-Packard Enterprise in 2016. And we actually partnered because they were among the first to develop memory-driven computing. So what is memory-driven computing? Memory-driven computing is a computer system which sort of does not utilize ROM and RAM memory to store data and put them in cache and fragment, but every type of processing occurs in the same memory at very high speed with photonics, so there is less connection, less copper, less energy consumption, very high speed. So the first challenge, it was to find out if memory-driven computing could help us, for example, in genomic sequencing. And what we discovered is that if you take a benchmark, which is about 140 million reads out of a sequencer, normally the process takes about 22 minutes, now we do it in 13 seconds. So this actually enables us to be able to process simultaneously 80,000 human genomes. And of course increases tremendously the capability of each single computer to be able to perform this data. How does this help us in sharing data? Well, if you really look, we have a number of those data which are generated: genomic, clinical data, laboratory data, imaging data, which are very heavy also in terms of sizes. And we want to integrate this data to use machine learning neural networks to visualize this data. And of course, the issue is going to be that we have to share them, for example, at different sites within the ZNE, but it could be, for example, here with Moscow or with the United States or with Japan. So we need to be able to share the data, and of course we have to respect the European legislation that patient data especially, and especially genomic data, cannot be put on the cloud because there are a number of regulatory mechanisms. How do we do that? Well, we are trying to do that together with HP by using swarm learning. So automated swarm learning is a way by which on the cloud, the only thing that you share is a code, is actually a container, is a box, which then is moving fast from one place to the other, delivering the modules and the software, which enables locally the manipulation of data and nearly instantaneously going back on the web and share the learning that has been taken place at that particular site with the learning that has been taken place at another site. So now by doing this kind of swarm learning, you automatically share the data and create complex analysis without moving the data from the patients from where they are. So we are actually quite advanced with this because we are planning now for the beginning of next year the first experiment in which we try to analyze simultaneously a vast number of genomic data at 4 different sites at DZNE. We're also collaborating with the Center for Cybersecurity at Helmholtz to be able to write the code that will protect also this system from, of course, to be able to protect privacy. And of course, in this way, we can implement services for research based on a container and not necessarily on the data themselves. So we have the data on the edge, and we just do the computing into the cloud, to the core. Everything goes back to the core and comes back to the edge without touching the data. So that's actually something where we are trying to be able to make progress significantly, to be able to share data. And this is an open source code, which means everybody can access it. So everybody can acquire the computer system from HP. They're not yet commercially available. They will be very soon. So then we hope that this will be then a way by which we can share human data very effectively between different sites. Finally, and I'm sorry this is not very eligible, but there was an independent research evaluation from Elsevier that sort of made us very proud because after 10 years of life, actually, we are linked among the top 10 organizations in the world doing research on neurodegenerative disease, actually among the top 3 to 5, if you look at the impact of scientific publication and cooperation with academia, with industry. So it is possible from a very short period of time by bringing together the very best brains within a country to be able to be competitive worldwide. So why not doing it internationally? And so that's my message in the end, and it's with this that I would like to thank you.
I think that maybe 1 or 2 short questions, if you don't mind. Sure. Short questions. Otherwise, we leave for discussion. Okay? No short questions. Well, let's go on. The second talk will be given by Professor Illarioshkin, Clinical Research of Neurodegenerative Diseases in Russia.
Dear colleagues, dear friends, good afternoon. In my short presentation, I'd like to characterize very briefly the current state and the main results obtained by Russian scientists in clinical studies of neurodegenerative diseases. And in our opinion, most promising problem in the field of clinical studies of neurodegeneration are indicated on this slide. These are biomarkers, risk groups, early diagnosis, neuroimaging and cerebral rearrangement in the course of neurodegeneration, of course, genetics of neurodegenerative diseases, translational neuroscience from experimental studies to clinics, and artificial intelligence in clinical neurology. And to begin with, I'd like to briefly characterize first topic. This is biomarkers and risk group and early diagnosis. As all we know, of course, one of the most important characteristics of neurodegenerative diseases is the presence of very, very long preclinical stage, latent stage, which may last from years to decades. And the main problem is to diagnose the disease somewhere around here in the time of conversion from preclinical to clinical studies, and even much earlier in the course of neurodegeneration. Is it possible or not? Yes. Now it is possible with less or high probability, and we use for that purpose the ideology of biomarking. So clinical, neuroimaging, neurophysiological, molecular genetics, including proteomics, genomics, and other pathobiochemical biomarkers. I'd like to show you this approach just as an example. In our studies on Parkinson's disease, one of the classical neurodegenerative diseases with a long-standing preclinical period. And you know that we have several biomarkers in Parkinson's disease, like hyperechogenicity of substantia nigra, of course, REM sleep behavior disorder, genetic biomarkers, some neurophysiological markers involving color vision or identification of alpha-synuclein in peripheral tissues. And we created a two-stage system in our country to screen patients for Parkinson's disease among healthy population. I'd like to show you the very first result of these studies. Let's see here. On the first step, we screened normal population for the presence of hyperechogenicity of substantia nigra or mutations in main genes for Parkinson's disease. So we formed on the first stage group 1, group of primary risk of Parkinson. Then we supplemented this group with additional neurophysiological biomarkers, a lot of them, which I've showed previously on the slide. And those patients, those individuals, healthy individuals, carrying one of the primary and several secondary biomarkers, so composed group of high risk of Parkinson's disease. So we followed this group for 7 years, and now the very first result to show you. You may see that we found increase in the number of biomarkers in the risk group shown here, and more importantly, several patients already from the group of high risk already turned out to have Parkinson's disease very recently. 4 of them are already definitely ill, and several patients with different probability of Parkinson's disease. Maybe we'll come to Parkinson's later. And nobody in the group of low risk of Parkinson's disease. So this slide principally show you the possibility to create the system for screening for Parkinsonian population. And of course, we'd like to supplement this system with some additional biomarkers of high validity. And transcriptomic studies seems to us the most promising in this field. So we perform transcriptome analysis in our patients with Parkinson's disease. And in parallel, we perform transcriptome analysis of animal models of Parkinsonism using 6-hydroxydopamine and other neurotoxins. And once we reconstructed genetic networks and biochemical pathway, we found several common genes involved in pathogenesis of early non-treated drug-naive Parkinson's and animal models of Parkinson, which those genes, those molecular products might be a good target for neuroprotective treatment. These studies have been published largely by our groups. And second direction, neuroimaging and cerebral rearrangement in the course of neurodegeneration. Of course, current possibilities of neuroimaging technologies are very impressive. So we can follow rearrangement, reorganization of the cortex of subcortical area from the very latent, very early stage of Parkinson's disease or Alzheimer's disease or any other diseases to the advanced stage. And again, several examples in this field of neuroimaging. For instance, here, at the very debut of Parkinson's disease, before any treatment, we can show already the decrease in spontaneous neural activity in the right lower parietal lobe, shown here. It's very important biomarker. And then, upon initiation of therapy, we can see some changes, increase of numerous of neuronal activity in many regions over there, shown here, which may also help us to understand the logic, the internal logic of brain rearrangement upon the course of neurodegeneration. And of course, you may see here another example on frontotemporal dementia. You may see here the striking decrease in the cognitive area, which is responsible for cognitive coordination, so-called, in the very early stage of frontotemporal dementia. And another methods, not resting state functional MRI, is here, but voxel-based MRI showed you, for instance, in the very early stage of tremor-dominant form of Parkinson's disease, the increase in the volume here of the thalamus, one of the structures which is relevant to tremor, and in a healthy person carrying the risk gene for Huntington's disease, you can see at the very preclinical stage a clear atrophic changes of the striatum, the main target for this disorder. And an important supplementation to the MRI is the technology of navigated transcranial magnetic stimulation. And so several studies has been published by researchers from our center in this field. This is a theory of method which allows us to finally map the muscle, hand muscle, for instance, into the cortex. And here is a healthy volunteer. This is representations of motor area used by transcranial magnetic stimulation with MRI. This is patient with amyotrophic lateral sclerosis. You may see the striking reduction in the size, in the square of the cortical representation of the hand muscles, which is very clearly seen here and correlated with the severity of the disease. Moreover, in a typical case of amyotrophic lateral sclerosis, without clear clinical involvement of the upper motor neuron, you can still see this sign, which can be helped in clinical diagnosis, in differential diagnosis, in the monitoring of pathological process, including the monitoring on the course of treatment. Next direction is genetics of neurodegenerative disease. Needless to say, in this audience, that genetics is very important in neurology, but especially in neurodegenerative diseases because of striking genetic heterogeneity of these disorders. As you know, all of us, one phenotype can be caused by not just tens, but thousands of genes. One phenotype, for instance, epilepsy and some other phenotypes. So we definitely need to use modern genetic technology screening like NGS in the exomic, panel variant, MLPI technology, real-time PCR, any technology which allows us multi-parallel analysis of genetic variants. And in our country, in collaboration with a lot of institutions in our country, in university across Russia, and in collaboration with our colleagues from different countries, we filled up genetic map from the viewpoint of all main group of neurodegenerative diseases: genetic structure of Parkinson, autosomal dominant and recessive ataxias, primary dystonias, Parkinson motor neuron disease, and so on and so forth. I'd like to stress that this works could be done only in collaboration with a lot of institutions, a lot of publication by our group, but there are several very interesting findings specific on Russian population. For instance, I'd like to stress that more than 10 genes and genetic loci have been identified for neurodegenerative diseases by Russian scientists, by Russian neurology. For instance, X-linked and progressive cerebral hypoplasia, a typical form of recessive progressive muscular atrophy, turned out to be dysphorinopathy. It's our groups from Research Center of Neurology. 3-form Charcot-Marie-Tooth disease identified by our colleagues from Research Center of Medical Genetics, and so on. Of course, we identified specific genetic features, characteristic of Russian population for neurodegenerative diseases, like, for instance, identification of the largest in the world cluster of spinal cerebral type 1 in Yakutia, more than 500 cases, which allowed us to study homozygosity for expansion and many, many other features. A number of population-specific mutations identified, and population-specific features of microsatellite expansions. It's a specific type of mutations characteristic of neurodegeneration. I mean, dynamics of intergeneration expansion, and so haplotypes, origin of this trinucleotide or pentanucleotide repeat expansion diseases, and so on. And as I mentioned already, international collaboration. It's a nice paper published this year in Annals of Neurology, in which there are 2 persons from our center participating. This is just an example of this collaboration international. There is a global team studying genetics of Parkinson's disease, and we collect families and individual cases carrying one or another mutations in different genes for Parkinson. And on this map in the world, there is Russia and 1 Russian center here in Moscow. This is Research Center of Neurology. This is just an illustration how many interesting findings can be supplied by some kind of collaboration across not only just Europe, across the world. Next direction, translation neuroscience from experimental studies to clinics. So when we are talking about clinical studies, of course, we cannot differentiate purely clinical or purely basic research. That's impossible. That's why it's very important to maintain some level of translation from basic studies to clinics. Of course, we have many examples of these studies, and I'd like to show you some examples on cell reprogramming, on studies based on induced pluripotent stem cells, because in this field we collaborate between just maybe 8 different institutions in different cities in Russia, for instance. Let me show you. Just briefly, for those who are not very familiar with this technology, so we can get any somatic cells and reprogram these cells induced pluripotent stem cells, so-called pseudo-embryonic stem cells, using several peptide factor of pluripotency, which can be introduced into cell by different ways. And this is a famous discovery by Japanese scientists who got Nobel Prize winner for this discovery. And in our studies, we used cells obtained from patients with Parkinson's disease and studied dopaminergic neurons. We obtained cells from patients with Huntington's disease, studying GABAergic spiny neurons, which are primary target for pathological process Huntington, and also spinal motor neurons from patients with ALS. And you may see here, we got specific neurons for each disease as confirmed by specific staining with immunohistochemical markers shown here, and also published in different papers. And I'd like to show just one example of our studies on Huntington's disease. As you may see here, once we get fibroblasts from patients with Huntington and transform them into the GABAergic spiny neurons specific for striatum of patient, we can see on these cell models specific finding characteristics of Huntington's disease: clear indentation and defects in nuclear membranes, defects in mitophagy and autophagosome formation, you may see here. Moreover, we with our colleagues from Institute of Cytology in St. Petersburg discovered the defect in so-called STO-operated calcium channels, relatively new type of calcium channels. And moreover, we showed that when we use one of the new compounds shown here, we can restore calcium channels through these, in this cell model of Huntington's disease, which clearly indicate this compound might be regarded as a new potential candidate for new drugs in not only Huntington, maybe in some other trinucleotide repeat disorders, at least of polyglutamine type. And also this data has been published in several papers, and I'd like to show you the main direction this field are: studies of basic mechanisms of neurodegenerative diseases, personalized approaches to the treatment, personalized because we can try one another drug in a specific neuron from one or another patient who was the donor of fibroblast. Screening for new potential drugs, as I shown already, correction of genetic defect by CRISPR-Cas9 system, and according to our experience, when we correct mutation in Parkinson's disease, we have such an experience, we can improve the phenotype of neurons carrying or not carrying target mutation. Experimental aspects of neurotransplantation, we have also some experience in transplanting those cells to animals with Parkinson's disease model. As you know, 1 year ago, first patient in Japan have been operated. We hope in our country, while completed our studies, experimental studies in our laboratory, to be able also to proceed to the transplantation of dopaminergic neurons obtained from patients from his fibroblast as a cure for Parkinson's disease, and of course, iPSC banking aimed at further basic and clinical studies. So these works have been done with our colleague from Novosibirsk, from St. Petersburg, from several institutions in Moscow and in other cities. And last direction is artificial intelligence and clinical neurology. Very interesting, very new, young, but very promising technology. And I'd like to show as well several examples from our own study. And first of all, let me show that we, our group, our research center, is a member of Global HD Studies through Enroll-HD, a study which was transformed from registry study. So you may see Russian-speaking site, Moscow, and several other cities which are working under the auspices of our center. We are coordinating center. So, owing to Enroll-HD database, so more than 8,000 patients already registered and studied from the viewpoint of clinical characteristics, we have access to this database because we are a very active participant. And owing to this access to unique database, we could able, using method of machine learning, to recalculate the curve which show the correlation between number of trinucleotide repeat in Huntington's disease gene and age of onset of Huntington's disease patient. This curve is much more relevant, much more exact than the old curve used by our colleagues, and show more clearly the correlation within the narrow range of very small number of CRG repeat, which was quite difficult to predict by old models. And another part of this study, so we were able to calculate the risk of suicidality in those carrying mutations Huntington's disease. As you know, suicidality is one of the major cause of death in patients with Huntington's disease. So we show that our mathematical algorithm can correctly identify that 40% of our patients can be at risk, at high risk for suicidality by the end of the second year of observation. And that's a very important point from practical viewpoint. Another very recent example, so with another group of mathematicians in our country in Parkinson's disease. So we use this portable accelerometer, long-term record of motor activity of patients, not only tremor, but any motor activity, wherein here on the hand, and several technical characteristics of algorithm use it, and we were able to show that with 90% of accuracy, this machine algorithm can discriminate correctly patients at a very early stage of Parkinson's disease from health control. Moreover, when we deduct those, when we remove tremor as the most confounding symptoms influence the curve, the validity, so the accuracy still remained at 90%. It's very high. It's very interesting to use in clinical practice further. We are going to continue these studies. And also, we have some experience with our colleagues in the field of early diagnosis of multiple sclerosis. And here you may see that machine learning results of diagnosis are very close already just today, very close to expert opinion. And our colleagues from St. Petersburg, Psychoneurological Institutes named after Bekhterev, are engaged in the project of prediction and conversion of mild cognitive decline to dementia using machine learning. And this is very important in early diagnosis of Alzheimer's disease. Well, so I mentioned a lot of institutions, a lot of cities. I'd like to show very briefly some results from our colleagues also from Krasnoyarsk State Medical University, because also translational neurology is well developed in these institutions, from original model of brain-blood barrier and target modulation of neuromuscular units, and studies of local microenvironmental in modeling neurogenetic disease to some neurorehabilitation technologies and the virtual reality and some robotic techniques and so on. Finally, I'd like to show several of our colleagues in Germany who are already in contact not only with our research center of neurology, with some our colleagues from Novosibirsk, Krasnoyarsk, St. Petersburg. That's not the whole list, of course. Some most active groups and some fields of studies indicated here. And of course, we would be happy, owing to our today's meeting, to be able to expand our collaboration with our German colleagues or our European colleagues in the field of growing, with the fields of dynamically developing field of neurology as neurodegenerative disease. Thank you very much.
Thank you, and we will leave questions for discussion. So the next talk will be given by Katrin Amunds, Fundamental Brain Research in Germany and the European Human Brain Project.
Can we dim the light again, please? This was very nice. Thank you.
Свет, пожалуйста.
So my talk will address more the basic scientific aspects, but I do not pretend to speak about whole Germany basic research, but rather I'm focusing on those German researchers who are involved in the context of the European Human Brain Project. So I would like to start my talk with the notion that the brain is a very complex organization that is organized on different scales in space and time. And we heard the perspective now of patients by Pierre Lucci. He started that, and of course the brain is a complex organ that is governing most of these processes. It is about 1-2 decimeters, but then it is composed of different structures and cortical areas, subcortical nuclei like the hippocampus, and again, very important structure also for neurodegenerative disorders. The hippocampus has different layers which have a different distribution of nerve cell bodies. And this implies that these different regions in the hippocampus have a different connectivity and function and contribution also to the disease. And when we think about connectivity, then we have an ill-posed problem because cells are very small, but they can have connections over several centimeters long. And the transfer of information is realized at the neuronal level at synapses, which are structures of a few micrometers large. And in these synapses, we have vesicles, and these vesicles are filled with neurotransmitters, and these neurotransmitters bind to certain receptors, and receptors, of course, are very typical target molecules for drugs that we are developing. And here we are arriving at the nanometer scale. So in my view, in order to understand the human brain, it is really important to bridge the different levels of brain organization. And this is probably also one of the most challenging aspects, because we are researchers in neuroscience, we are working on a certain level or maybe on 2 or 3 levels, but it's very difficult, very hard to oversee the whole spectrum of activity. And that's make it so difficult to work alone in a lab and really requires to join forces. And the Human Brain Project was set up in order to join forces in human brain research with a 10 years horizon for funding. 1 million was the planned funding. Now we are a little bit past half-time, and we are looking forward to the last phase starting in 2020. The core project involves 116 institutions from 19 countries and 12 different sub-projects. There are 27 partners from Germany, so Germany is really one of the strongest partners in the Human Brain Project and contributing in different aspects. So we are at the interface of neuroscience, computing, technology, and brain medicine, and that makes us quite unique in the comparison with other international brain initiatives which focus more on either on one species or on one aspect. And what we are now doing is developing a research infrastructure called eBrains, because we want to create a sustainable platform for researchers worldwide to collaborate, to share data, and to use the tools that have been developed and to further develop it. And how do we do it? Well, we very much rely on co-design and use cases. What does it mean? Use cases mean that I, as a neuroscientist, have a specific research question, and together with engineers, informaticians, mathematicians, we are developing tools to address such types of research questions. So we call this a co-design project. Our mission is to understand the multi-level brain organization in space and in time, and to bridge the different levels, to understand also how a certain change at the molecular or genetic level is influencing at the end something complex like a behavior or evolving clinical symptoms. So we want to transfer this knowledge to brain-derived applications in health, computing, and technology, to contribute at the end that patients have a certain benefit from our research, to make sure that we translate our knowledge into new computing technologies, but perhaps also other neurotechnologies. And eBrains is our research infrastructure that should serve these aims. At the end, we want to create a benefit to the European society and ensure that this is an ethical approach in research. And this is challenging, as Pierluigi also pointed out. So we are facing quite some problems with sometimes contradictory assumptions going on the one hand to make data more transparent, available, annotate them as good as we can. And at the same time, we have to protect privacy of patients and of subjects. And this is not easy to combine in one and the same approach. So a central element of our endeavor is to develop a human brain atlas. And it's a little bit like Google Earth. So maybe it's our European answer to Google Earth. So we want to develop Google Brain, but made in Europe. And in a very similar way as we have a map of the Earth, we have a coordinate space of the human brain. We have maps of regions, like we have maps of countries in the world. And we have to invent a taxonomy with names and relationships between brain regions and different categories. And in the same way as we have different maps when we think about the Earth, reflecting politics, topography, or climate, we have the same approach in the human brain. We will have different maps. What you see here is one of such different maps, revealing the molecular organization of the human brain based on the distribution of transmitter receptors. And transmitter receptors are important molecules for signal transduction in the human brain. And what you can see here, that different transmitter systems like acetylcholine or glutamate or GABA, they do have different receptor types, and these different receptor types have a different distribution pattern and concentration locally in different brain regions, but also layer-wise or in substructures. And this is very interesting and relevant for us when we want to make new drugs for patients, because the binding patterns are so different independent where you are in the brain. And of course, this is not that's not the only prerequisite, but knowing better what is the distribution of these receptors will help us also to better understand the functionality of drugs that are acting on the brain. And just in terms of basic research, these different distribution of neurotransmitter receptors help to find out that Broca's region, which is a classical language region, is much more complex than we thought. And 150 years ago, the idea was, well, there's more or less one region, it has two areas, and there are a lot of neurology textbooks speaking about just two areas, 44 and 45. 10 years ago, we found out that there is more than a dozen areas probably in this region. And now we have a concept which goes towards 50 new areas. And that's not a question of, so to say, spatial resolution. It's also a question of different functionality that these areas have. And when we do understand, when we want to understand language, which is such a complex phenomenon, we have to understand the structural functional relationships, and we see that the whole universe, let's say, of language-specific areas in the frontal lobe. And the Wernicke region, of course, is not better, and I would suppose it's similarly complex. And we see that these different brain regions also have different types of function. Some might be more closely related to language, but others are, so to say, more in the field of gustatory or other fields. Maybe this is a question of our limitations with spatial resolution, but now we have a precise or more precise tool to test such hypotheses. So we will include more and more these data into an atlas, and you see here one template of the Human Brain Atlas, which is a freely available resource. It's a so-called big brain template, where we label different brain regions, and what we are now aiming to do is to go to the 1 micrometer range. But when we are at 1 micrometer, that would mean that we end up with data sizes of 2 to 3 petabyte. And there are no tools available at this moment to exchange such large amounts of data via web-based services. So we have to think about how to make this possible, again, under the GDPR and other constraints. So this atlas is only starting to be interesting, but still we have about 500 visits per day. And I think the more complete it is, the more it's interesting also to use this map, to download it. And it's not only including information about cytoarchitecture or cytoarchitectonic areas. We are including data about gene expression together with the Allen Brain Institute, or connectivity, or molecular organization. So it's a true multimodal atlas. So what it is good for? So one example from clinical application from my colleague Petra Ritter in Berlin, and Victor Josa, who is working in Marseille with epilepsy patients. So they are developing individualized models under the umbrella of the virtual brain for medicine and research. And the idea here is to help neurosurgeons to make better surgery planning for such epilepsy patients. And first studies showed that indeed, if the predicted if the by virtual brain predicted region where tissue can be removed is most closely related to the natural or to the real result of these patients, then the outcome is better. And it is so important to also contribute here because the benefit for patients after neurosurgery in epilepsy is not so much changing, although we are working very intensively. And Petra and her team are now translating this virtual brain also to cases of stroke, of tumors, but also interestingly to Alzheimer's disease, where they start to model the distribution again of neurotransmitters in these Alzheimer's patients. So these are kind of individualized models that could help to predict the outcome of surgery, but also, of course, to better understand what are the features of normal brain organization and structure. And when we think about exchange of data, I would also like to mention Simon Eickhoff's approach in our institute. So he's developing a decentralized data exchange infrastructure. And the idea is here that from the very first point where you scan patients or healthy human subjects, and then the data is being calculated, analyzed, maybe using high-performance computing storage services for analytics. And then at the end, we have a paper in our hand. So all this process is very carefully monitored, and we have a new type of provenance tracking. And again, I think this is a very important step because there's a little bit of crisis in neuroimaging and also in other fields because things are not reproducible. Why not? Because data are not well enough, good enough annotated. And we are here developing ways to make such neuroscientific experiments trackable and reproducible. And this is, in my view, absolutely necessary, not only to share the data, but also to annotate them in such a way that others can replicate. So we going also towards a better understanding about the network architecture in the human brain. And what you see here, these are tractograms illustrating what is the course of axons in the human brain and the hippocampus again. And this is extremely challenging in terms of compute resources, but also in terms of underlying mathematics. And we hope here to make progress, not only to be within the hippocampus, but of course the hippocampus is connected, and we would also like to understand long-range connections on the axonal level using this method. So such network models will help to improve neurobotics, and we have a group in Munich led by Alois Knoll, who is doing innovative robots and brain-inspired simulation. So the idea here is not only to develop physical robots, but also to develop simulation of robots, which is a little bit cheaper at the end, and which allows under very well-described conditions to perform experiments. And for example, visual guided movements, this is a perfect model where I think also industry can benefit quite a lot from learning more from the brain, how the brain is coordinating vision and motor. And another technology application is so-called neuromorphic computing. We have in the Human Brain Project two systems, Spinnaker and BrainScales. Both are now further being developed in Germany, one in Dresden, this is Spinnaker, and Heidelberg is famous for the BrainScale architecture, which is a spiking neuromorphic system, and it seems to have certain advantages if we want to model and simulate learning and plasticity in real time, which is still a challenge for classical new supercomputers. So we are also moving along this direction. And the Spinnaker, I mean, these are very small chips, and it's interesting to put them into robots again, or use them when the energy consumption problem is a very important one. So again, I see here that we can learn from the brain and also use it for brain research at the end. So we do have a lot of data, and we heard it already several times. And when we just compare rat, vervet, and monkey data, how many terabytes or petabytes we have, we are running, of course, into a very large data handling problem. And it's not only the problem that's a huge amount of data, it's also the heterogeneity of data. So genetic data are completely different from physiological data, are completely different from neuroimaging. Each community has its own regulations, data formats, and ways to annotate the data. So it's really, data science is one aspect that we should keep in mind. And in Jülich, we are developing not only high-performance computing systems of the future, but also data science center of the future. And Thomas Lippert will tomorrow show more, of course, but for those who cannot be present there, I would like to mention that he is developing a modular type of supercomputing, and the idea is that there is not the typical neuroscientist, or there is not the typical genetics person. Yeah? We all have workflows that are very complex, and we are developing in Jülich a system that has a cluster, that has extreme storage components, data analytics models, perhaps in the future also include more the quantum annealer, neuromorphic devices, or the booster when we want to have really high scaling simulations. And we see that different communities use these modules in a different way. So climate research, of course, needs this cluster booster architecture, which is already running, and which is, so to say, allowing to simulate at high scale. But when we think about deep learning, then perhaps neuromorphic systems with a cluster architecture are better suited. Or when we think about HPP analytics of data, then we need extreme storage, at least I need it. And we need the data analytics model and perhaps a quantum annealer when we think about certain types of problem in the future. So if we would have a system that allows, so to say, seamlessly without the intervention of neuroscientists who have no idea what is going on in the background, if we have such a system that, so to say, makes these computers adaptable to the needs of a certain scientist, this goes, of course, far beyond neuroscience. It's also important for much more research fields than we can think now about. And to make this available, we are building a European digital infrastructure at this moment with 5 supercomputing centers. Jülich and Lugano, CSCS, have already very intensive contacts. And contacts means that we in Jülich, for example, calculate certain analysis. And then when we have the result, we put it to CSTS, and they have, so to say, the web-based access for a certain deep learning network. And we would like to further include Italy, Cineca, the BSCS in Spain, and CEA in France, so that we can really use also the advantages of the different computer architectures in the different European countries. So that's a little bit the idea of how the Human Brain Project would like to continue. And we have developed, and this is my last slide, a nice exhibition, I think a very nice exhibition, which was just opened a few weeks ago at the Bundestag in Germany with leading politicians in Germany. And this exhibition should serve, in my view, as an ambassador of neuroscience to politicians. And I'm wondering whether Russia, whether the Duma would be also happy to have such an exhibition where we can explain people how important it is to foster brain research, not only because we want to understand the human brain. Of course we want it, but because also we want to help patients at the end and also support our society and economy. Thank you for your attention.
Thank you, Catherine. Is it exhibition movable? You can bring it to Duma?
The concept was such that we can transport it and we can put it into one big car and that's it, and put Russian or English letters to it. So that was the whole idea. That's a traveling exhibition through all Europe.
I think it's worth to think about Duma. Thank you. So the next talk will be given by Professor Anokhin, Fundamental Brain Research in Russia.
Dear colleagues, ladies and gentlemen, it is virtually impossible to cover the whole field of fundamental brain research in Russia. I will not address such important and traditional questions as mechanisms of synaptic transmission, characterization of different types of receptors coming from the work of Samoilov at the beginning of the 20th century and going on at various institutes in molecular biology, genetics, and neurophysiology, not sensory physiology, not motor physiology, not traditions in the neurohistology and anatomy from different schools, so in Kazan, St. Petersburg, and Moscow. I will focus on one main trend in fundamental brain research in Russia. It is neuroscience of higher brain functions. The reason I am going to focus on this is twofold. First, by the majority impact, since this field has been the major field in physiology, and if you look at the Society for Physiology from the 20th century up to now, there are huge sections of physiological society and meetings which is devoted to higher brain functions. Actually, Russia has been holding probably the longest and largest brain project, started from 1920s and up to 1990s, disappearance of the Soviet Union, which was devoted to the studies of the higher nervous activity in the various aspects, clinical, psychological, neurophysiological, comparative, etc. And the second reason is that despite the huge amount of big data which is accumulating now in neuroscience, it is not possible to squeeze out of this big data the mind and consciousness. The major discoveries in this field are yet to be made. And in a sense, the traditions in fundamental brain research in Russia are going from the past and the focus on this question into these future questions and discoveries. There are three features of Russian neuroscience. I will not present many facts in my short presentation. I just want to leave you with a certain feeling or impression of traditions and current research and flavor of research in neuroscience of higher brain functions in Russia. The traditions or features are that from the very beginning of the brain studies in the 19th century, mind has been a main focus for neuroscientists in Russia. They were taking a materialistic approach to this. They were treating the brain as an organ of mind and looking for a physiological basis of mind. And we have generally in Russian science and in this field the tradition of scientific schools, which is important in addressing complex questions like that through several generations. So what I will do now is that I will go briefly through these past traditions, then we'll describe the landscape of present neuroscience research, taking as an example calls and programs which are funded presently or more recently by Russian science foundations, which shows the priorities in the fundamental brain research in this area. And finally, we'll say a few words briefly about the project of the program of research for the years coming up to 2035, which is under consideration, and how neuroscience and brain studies is represented here. And I will finish with a small example from our main work, just giving the practical impressions from these three areas. So focus on mind. Russia has a cultural tradition of addressing the issue of higher brain functions through the physiology. Ivan Sechenov, who made the major experimental discoveries in the central inhibition and plasticity of the nervous system, was first to pose among the physiologists in the world the question of physiological studies of the basis of mind. He has been known as well as a psychologist, as a classical neurophysiologist, and 19th century discussions of Tolstoy, Turgenev, Chernyshevsky are focusing on the ideas which were brought to Sechenov, by Sechenov into the Russian society and culture. It has influenced also next generation of neuroscientists in Russia, like Bekhterev and Pavlov. Bekhterev was starting from neuroanatomy, coming from the famous Kazan neuroanatomic school. It's interesting that the Kazan Museum holds the first probably 3D reconstruction of human connectome made personally by Sechenov after tracing the pathways in the human brain. But his focus was also into bringing the ideas of how the mind is operating in this brain. It is probably less known that Bekhterev was co-discoverer with Pavlov at the same time of what is called conditioned reflexes, and he called that conjunction reflexes, independently developing the research of these at the beginning of the 20th century. Of course, Pavlov has brought experimental techniques to the study of the physiology of the higher nervous activity. He was genuinely interested in the experimental studies of what he called the molecules of mind, the links between the associations between the various behavioral acts. And starting from 1913, he started to speak about the physiology of higher nervous activity, which was then transformed into a major trend of research in Russian neuroscience. Actually, we do not have what is interesting at our universities. We do not have neuroscience departments. We have, at the biological faculties, we have departments of physiology, and we have the departments of higher nervous activity, where the neuroscience is taught, but mainly with the aspect of the higher brain functions. The difference between the Russian tradition in approaching this question and Western European tradition is summarized in this exchange of opinions between Pavlov and Sherrington. When Pavlov was visiting England at the beginning of the 20th century, Sherrington told him, "I don't think that your conditioned reflexes will have a success in England. They smell too materialistic." While Pavlov consistently was claiming that this is a way to develop the physiology of the brain. And into translation of the book Mechanisms of the Brain as a suggestion, he said, it contains nearly nothing of Sherrington, new except extravagance that our mind might have no relation to our brain. So this tradition continued into the studies of the higher brain functions using the electrophysiological techniques EEG, and the first approaches to synchronize brain activity were developed in Russia. Actually, the IBRO, International Brain Organization, was conceived and initiated in Moscow in 1958 at the colloquium on EEG and conditioned reflexes, international colloquium. Russia also initiated and holds probably the largest international brain research program. It was started in 1960 and lasted for 30 years until the end of the Soviet Union in 1990. It was called Interbrain, and it cooperated the research in neuroscience in the 15 republics of USSR and 9 countries of the socialist camp, so 24 independent countries now. However, in the 1990s, when the revolution in techniques of neuroscience, in human brain imaging, in the cellular and molecular biology techniques developed in neuroscience, we had difficult times. So presently, we do not probably have the sufficient amount of modern technology and equipment, but we have this large tradition. There are about 100 research institutes, universities, and national research centers are carrying out active neuroscience research in Russia. They are concentrated mainly in the western part of Russia, in Moscow, in St. Petersburg, in Nizhny Novgorod, in Rostov, in Kazan, but also in Novosibirsk, Krasnoyarsk, Vladivostok. To give you a feeling of what are the priorities of current neuroscience research, I will take an example of a recent program, a priority program from the Russian Science Foundation. It is a major science foundation which supports large research projects in Russia. The call was, and this characterized again the approaches and tradition on neurocognitive research and technologies, and was divided into three topics. One was fundamental mechanisms of brain and cognitive function, so higher brain activity or higher cognitive functions, which included funding the projects in the field of brain mapping, neuroimaging, neurophysiological studies of human cognition, the search for new targets for drug development, inquiry into the cellular mechanisms of information processing and storage. There is a large tradition in neuroscience or in Russia on learning and memory at different levels. Establishment of neuroarchitectural principles of cognitive functions, including also functional neuroimaging and connectomic analysis. And mathematical modeling of neuron encoding of cognitive information using the traditions and available groups in mathematics and theoretical physics in Russia. The second topic was new techniques to develop the study and modulation of cognitive functions, and the projects were in the fields of development of methods and software for analysis of neurocognitive data, computer atlases, digital brain atlases, databases, analysis of images, development of new methods for modulating neural activity and brain functions, including novel techniques in hemotherma and optogenetics, electromagnetic and ultrasonic modulation, non-invasive brain stimulation. Prediction of human behavior through analysis of neurophysiological parameters, and technologies for neural interfaces which are developing you have seen some of this research today at the Center for Neurology, which are developing both for non-invasive interfaces and also implantable neural interfaces, including bidirectional optical fiber neural interfaces and optogenetics. And clinical research and humanitarian studies, including neurocognitive studies of mental development and acquisition of knowledge into educational fields, clinical studies of cognitive functions and their disorders, using the disorders to understand the cognition, and neurocognitive studies and research in social humanitarian disciplines. Now, if we will look into the future, as I mentioned, there is a coordinated plan of fundamental scientific research, which is coordinated and has been prepared by the Russian Academy of Sciences. And there, neuroscience is present in several chapters, sections. In biological studies, where there are both molecular studies and studies of the development of the nervous system and ecology, mainly in section of physiological studies and sciences. And you can see, again, there is a major chapter on the higher nervous activity, neurophysiology, and physiology of cognitive functions. Medical sciences, again, the focus is on integrative basics of brain activity and in normal and pathological conditions. And in social studies, this is also cognitive neuroscience studies. Just to finish and illustrate how these things are coupled together, I will use an example from our own studies. We started as a tradition from the past, from the ideas developed in 1930s about the synchronous brain activity performing a functional activity with a distributed localization. Using the new capabilities of immediate early gene expression induced by neural activity, we developed techniques for mapping these functional systems globally in the brain. Initially, we were doing this with serial sections of the brain in animals with a cellular resolution mapping, but at the beginning of 2000, we moved to the Transparent Brain project. We developed techniques to optically clear the whole brain of the experimental animals, preserving the fluorescence from the transgenic proteins expressed under promoter of the genes, and to develop microscopic techniques with the light sheet microscopy with the physicists in collaboration, which allow to image the whole brain with a cellular resolution, looking for the ensembles of brains activity in a particular cognitive task. Why I bring this particular example for our today session, because it relates also to the issues of neurodegeneration. What we have found with this mapping in 1990s are two important facts. First, that during the retrieval of memory, there is a reactivation of plasticity genes in the brain. And if to prevent this activity, not being capable, for the brain not being capable to remember the retrieved old memories, it is possible to damage the old memories. We are starting now a project which is based on this data and suggestion that the reason for deterioration of memory in dementia, and especially in Alzheimer's disease, is due to this active process of self-degradation of memory under the conditions of the brain not being able to normally consolidate long-term memories. So whenever there is a retrieval of memory under conditions when it cannot be reconsolidated, the trace of memory is weakened and degraded. This leads to completely new ideas of how to protect active memories, comparable to the passive decay of memories due to synaptic loss. And another thing which we found is that with the systems mapping of the cellular activity, we see that each episode of new episodic experience is represented in a huge population of hundreds of thousands of neurons, which is virtually impossible to kill or to destroy by neurodegeneration. So what we have found in the experimental damage of memory is what happens is that majority of the cells which were involved in these experiences still retain memory. They are losing the ability to be integrated together. So the different parts of the brain can respond in an isolated manner to the sensory stimulus, but animals show amnesia. And we also found the techniques with a reminder which can reintegrate the circuitry. So to return the lost memory back, it is a long-term process. It takes several hours in different species of animals. It is NMDA and protein synthesis dependent. And we think that this can be a promising approach to restoration of memories which are still retained under the neurodegenerative conditions. Thank you very much.
Thank you, Professor Anokhin. Are there any questions to Professor Anokhin? No, thank you. Dear colleagues, now we'll have already completed our official program of presentations, and we have time for coffee break. And I think we will use that time to discuss all questions which can which can appear after our official presentation time. Thank you very much, and I would like to invite you to coffee break.